In this session, our guest speakers will be discussing the presentation titled "The Power of Whole Exome Sequencing to Unravel the Cause of Rare Disease." Our first presenter is Dr. Kraft, who studied biochemistry and molecular biology at Friedrich Schiller University of Jena, with his diploma thesis and PhD focus on cancer genetics. He held a postdoc position at the Institute of Immunology of Jena, where he investigated mast cell signaling, and had a postdoc position at the Institute of Human Genetics Aachen, where he investigated the molecular cause of rare inherited diseases by whole exome, whole genome, and nanopore sequencing and basic molecular techniques. Our second guest speaker, Dr. Begemann, studied biology at Heinrich Heine University, Germany, and RWTH Aachen. His PhD thesis was in human molecular genetics and epigenetics, with a focus on imprinting. He has studied at the Institute of Human Genetics University Hospital, RWTH Aachen, where he was also a postdoctoral researcher. I would like to thank both speakers for their time in putting together today's presentation. We will now begin today's session.
Okay, then thanks for this kind introduction, and thanks to IDT to giving us the chance to present our work here. We want to talk about the power of whole exome sequencing to unravel the cause of rare diseases. But I want to first start where we work. So we work in the University Hospital in Aachen. It's a very special building. You can see it, all these pipes outside. It looks more like a factory, and it's only one of two buildings worldwide built in this architecture style. One is the Centre Pompidou in Paris, France, and the University Hospital in Aachen. So if you've ever been in Aachen, step by and make your own impressions of this unique type of building. But now we come to the scientific part. So the research of our institute is focused on the pathomechanisms behind rare or ultra-rare genetic disease. So we started more than 6 years ago with whole exome sequencing. At this time, there were not so much enrichment chemistries on the market. So we started with Illumina, which was good. However, we had some problems that some genes are not covered or not fully covered. So we tried also different chemistries. And 3 years ago, we tested the IDT xGen Exome Research Panel oligos together with the Illumina enrichment and library preparation chemistry, and see that it really improves our enrichment results. However, short after the initial test, IDT came up with the Lotus DNA library preparation kit and also a hybridization and wash kit. So we fully moved to IDT reagents for our whole exome sequencing pipeline. And this gives us some advantages. So we have now most of the genes covered 100% at 20x. We have a more uniform coverage, which allowed us to include more samples per flow cell. We have higher flexibility with the spike-in probes, but I will tell you later on more on that. And it's a one-part protocol for library preparation, so you can easily add all the reagents to one tube, which is, of course, better for manual handling or also for automation. But now we want to show you some of our new disease genes we could discover in the recent years. And I want to start with a patient which came to us with a general dystrophy and a developmental delay. He also shows liver dysfunction, brain abnormalities like hypomyelination and also an atrophy, a muscle weakness, and some immune-related phenotype like an arthritis and autoinflammatory signs. And putting all this together, there was the hypothesis that it's perhaps mitochondriopathy. And when you look for mitochondriopathy, of course, you have to check the mitochondrial genome. And this could be easily enriched by spiking in the xGen human mtDNA research panel into the exome oligogroups. And as you can see, we tried different amounts of these research panel spike in, and this correlates with the coverage you get for the mitochondrial DNA. So if you want to look for a low copy number alterations in the mitochondria, you can use a higher amount to get a more depth in the coverage. However, when you want to have a general view on the mitochondrial DNA, you can use less what you need. However, in our patient, we couldn't identify the pathogenic mutation in the mitochondrial DNA. So we checked the nuclear genes, and we could find a homozygous loss of function mutation in the so far functional undescribed gene C2ORF69. And with the help of existing collaborations and also GeneMatcher, we identified further 4 families with additional 7 patients also having loss of function mutations in C2ORF69 and showing a very similar phenotype, clinical phenotype. And because the gene was functionally not described, we also do some functional studies. So we want to know where C2ORF69 localized in the cells. So we co-expressed an EGFP tagged version of C2ORF69 with several cellular markers, and one of them was TOM20. TOM20 is a marker which localized in the outer membrane of mitochondria. And you can see on the fluorescent images that there is a strong overlap in the localization between TOM20 and C2ORF69. So we thought that C2ORF69 could be a mitochondrial protein, and that's why we checked also mitochondrial function. And one major function of mitochondria is the production of ATP via the oxidative phosphorylation. And we could see in our C2ORF69 knockout cells, which we generated, that 3 complexes, complex 1, 2, and also 5, show lowered activity. So yeah, we could identify a new gene for disease for new mitochondriopathy. Another patient we had was a patient showing epilepsy and a muscle atrophy in combination also, which could be seen on the MRI image, thinning of the white matter and hypomyelination, and also in electromicrographs we can see that the myelin sheets of the nerve fibers are thinner than in control patients. And in this patient we can find a mutation in DEX1. DEX1 is a desaturase which is involved in the synthesis of sphingolipids. And we could identify a homozygous mutation, missense mutation, but we didn't know if this mutation can cause the phenotype. So we made some functional assays. And first of all, we can show that this mutation seems to destabilize the DEX1 protein. So we can see in comparison to the control that the DEX1 protein level is pretty low in the patient cells. And as I said, DEX1 is involved in the sphingolipid synthesis. So DEX1 introduce double bond into dihydroceramide, converting it to ceramide, and this could be further converted to sphingomyelin. However, if if the DEX1 activity is low or abolished, then sphingomyelin will be generated from dihydroceramide, so leading to dihydrosphingomyelin. And we could see this in the patient. So in compared to all the controls, C1 to C6 and the family, I can see a strong increase in these dihydrosphingomyelin. So this proves that DEX1 is really impaired in the patient, and this also explains, because sphingolipids are important for the myelin sheets, the phenotype we could see in the patient. However, if you want to discover new disease genes, it's of course important to exclude known sources of diseases, and these alterations are mostly in coding region, but sometimes also in UTR regions or deeper intronic regions. And these are normally not enriched with exome sequencing chemistries. However, IDT offer with the spike-in probes easy way to get all the regions in the genome you want in enrichment. So you just talk to IDT. I want this region into the enrichment, and they design and also synthesize the probes. And I have here one example. It's the JB1, a gene which is linked to a neuropathy. And there are deeper intronic mutations, also mutations in the UTR in these regions are described to cause a phenotype. And you can see in the normal enrichment they are not included. However, when we add some spike-ins, we can also enrich these parts of the gene. Another possibility to use the SPICAN probes is to get more coverage on difficult regions. I have one example here with selenon, where exome 1 is not good coverage. So we see without the SPICANs, we have only 6. We have minimum 6-fold coverage, which is not so high. So we included some additional probes to improve this, and now we have 41-fold as minimum coverage. And with this, you can make a reliable analysis if there are pathogenic mutations in this area or not. And with this, I want to hand over to Matthias, who will also present you now some of our projects.
So thank you very much, Florian. The first case I'm going to present you is a young adult that came to us for genetic counseling in cooperation with some other clinics in the hospital. And in her early childhood, she developed a sonic hedgehog medulloblastoma. And in the first diagnostic approach, the usual suspects or mutated genes like PET1, SUFU, smooth end could be excluded, and no causative mutations could be found. We then decided to include this patient into whole exome sequencing, and were able to identify frame shift mutation, a heterozygous frame shift mutation in the G protein coupled receptor 161, it's GPR161. And because recently GPR161 was described to cause medulloblastoma in mice, we got in contact with EMBL and made a cooperation, and they looked up their cohorts, and in this cooperation we were able to identify 6 patients in total with heterozygous dominating mutations in GPR161 that are listed here in the figure. They are almost all were loss of function mutations, and they were distributed all over the gene. When we looked at the age of onset, the patients with mutations in GPR161 had a similar age of onset like these patients with mutations in SUFU or PET1. And interestingly, you need a second hit in GPR161 to develop such a early onset childhood medulloblastoma. And in 5 of these 6 patients, the second hit was a copy number neutral loss of heterozygosity on the long arm of chromosome 1. That brings us to another interesting aspect of our exome sequencing. So we are not only interested in single nucleotide variants, but also in copy number variations and loss of heterozygosity that could be detected with the whole exome data. You need a comprehensive analysis to get this information, and therefore we spike in the xGen CNV backbone panel, which adds around about 9,000 additional probes outside of the coding regions with the mean distance somewhere around 0.34 megabases, and that allows you to have a more valid and accurate detection of CNVs. And to find these CNVs, we use the CNV software. This is an example of a deletion on chromosome 2. And as I mentioned, we're also interested in copy number neutral loss of heterozygosity. This is partly because we have a long history of researching imprinting disorders, and one of these imprinting disorders is Serratus syndrome that can be caused by maternal uniparental disomy of chromosome 7. That means that you only have maternal alleles of your chromosome 7 left, and this leads to the loss of paternally expressed genes, and finally to the Serratus syndrome, which is mainly characterized by short stature. And because of the SNP probes in the CNV backbone panel and the SNP probes in the coding regions of the exome, you can easily detect copy number neutral loss of heterozygosity or uniparental disomies like here on chromosome 7. Another interesting case that was published in August by our colleagues RWTH and Knop were loss of function mutations in the WDR11 gene, which are associated with intellectual disability and short stature. Yeah, so far heterozygous missense variants in WDR11 are associated with hypogonadotropic hypogonadism, or also called as Kalman syndrome. But so far, no cases with biallelic loss of function mutations has been described. WDR11 is involved in cilia function via the hedgehog signaling and endosome trafficking. And also, again, in a collaboration, we were able to identify 3 families with 6 patients that are affected by biallelic loss of function mutations in the gene. Their clinical features were mild intellectual disability, microcephaly, and short stature. And we were also able to stain the WDR11 in patient-derived fibroblast and control cells. And you can see here the immunofluorescent staining of WDR11 in green. And as a control, we also used Golgi 97, which is a trans-Golgi marker. And as you can see here, there's almost only background signal left for WDR11 in the patient-derived fibroblast with the homozygous stop mutation. And you can also see it in the merged picture that these Golgi 97 and WDR11 co-localize in the Golgi apparatus. We also try to quantify this, and again, you can see that you have only almost nothing left for the WDR11 signal in the patient cells, and also you can see this in the results from the Western blotting from protein lysates derived from patient cells. The last case I like to present is hereditary myopathy caused by mutations in the SVIL gene. SVIL encodes for the superbilin protein that is involved in myofibrillar assembly and interacts with costameric and sarcomeric proteins. And in so far, no muscular disorders have been associated with SVIL, and no mutation has been described. Again, in a cooperation, we were able to find loss of function variants in 2 families with 4 patients that carry again a Frank-Shift mutation and a nonsense mutation, and they had a very impressive wide neck and hypertrophy of the trapezius muscle and other back muscles. They had elevated creatine kinase and also joint contractures and stiffness of their fingers. And here we were able to perform electron microscopy and light microscopy staining in our department for neuropathology. And as you can see here, you have some protein aggregates and autophagic vacuoles, and also you can see the disrupted sarcomeres in the electron microscopy, and some problems with the membranes in the muscle cells. So they are quite disturbed. To sum up, we think that our whole exome sequencing is a great tool to discover new genes and new disease genes. Almost all of these genes would have been missed only by panel sequencing. Besides the possibility to identify coding variants, you also have the chance to detect variants in mitochondrial genes and also CNVs and loss of heterozygosity. We really appreciate the uniform coverage and the flexibility to add spike in probes and other to improve the targets, as Florian mentioned, and also that we have a one-part protocol at reasonable costs. And yeah, I hope we could show that that whole exome sequencing can be a great source of research projects, and yeah, can generate deep insights into the biology and contribute to scientific progress in our institute. With that, I have to thank, or we have to thank all the patients and the families, and of course, we did not do this all alone, but a lot of collaboration partners worked with us in Germany, here in Aachen, and worldwide. And of course, we have a great team here at the Institute that makes all that possible and supports us in all our research. So thank you to our team and to Florian and to IDT to give us the opportunity to present our work here today. Thank you very much.